Hello students, welcome back to Ligase Academy, your educational catalyst. Today we will be discussing about eukaryotic replication, elongation and termination. As I said earlier in the beginning class, the replication in prokaryotes as well as in eukaryotes are almost similar, but there are some differences. So come, let's get into the video. So, we have already discussed about initiation stage. A polymerase switching event was happened in initiation where primase was replaced by pole alpha, which is later on replaced by pole epsilon or pole delta. So during elongation, an enzyme called DNA polymerase adds DNA nucleotides to the 3' end of the newly synthesized polynucleotide strand. The template strand specifies which of the 4 DNA nucleotide adenine, thymine, guanine or cytosine that is added at each position along the new chain. Only the nucleotide complementary to the template nucleotide at that point is added to the new strand. So, DNA polymerase contains a group that allows it to bind to a single stranded template DNA and travels one nucleotide at a time. So for example, when DNA polymerase meets an adenosine nucleotide on the template strand, it will add a thymine to the 3' end of the newly synthesized strand and then moves to the next nucleotide on the template strand. So this process will continue until the DNA polymerase reaches the end of the template strand. So, DNA polymerase cannot initiate new strand synthesis. It only adds new nucleotides at the 3' end of an existing strand. So, all these newly synthesized polynucleotide strands must be initiated by a specialized RNA polymerase that is called trimase. So primase initiates polynucleotide synthesis and by creating a short RNA polynucleotide strand complementary to template DNA strand. So this short stretch of RNA nucleotide is called the primer. So once RNA primer has been synthesized at the template DNA, primase exits and DNA polymerase extends the new strand with nucleotides complementary to the template strand. So eventually, the RNA nucleotide in the primer are removed and replaced with DNA nucleotides. Once DNA replication is finished, the daughter molecules are made entirely of continuous DNA nucleotide with no RNA portion. So what is leading and lagging strand? So DNA polymerase can only synthesize new strands in the 5' to 3' direction. Therefore, the two newly synthesized strands grow in opposite direction because the template strand at each replication fork are anti-parallel. So the leading strand is synthesized continuously towards the replication fork as helicase unwinds template double-stranded DNA. And the lagging strand that is synthesized in the direction away from the replication fork and away from the DNA helicase unwinds. So this lagging strand is synthesized in pieces because the DNA polymerase can only synthesize in the 5' 3' direction and so it constantly encounters the previously synthesized new strand. So the pieces are called Okazaki fragments and each fragment begins with its own RNA primer. So, eukaryotic chromosomes have multiple origins of replication which initiate replication almost simultaneously. So, each origin of replication forms a bubble of duplicated DNA on either side of the origin of replication. Eventually, the leading strand of one replication bubble reaches the lagging strand of another bubble and the lagging strand will reach the 5' end of the previous Okazaki fragment in the same bubble. So Okazaki fragment maturation, it begins with the collision of pole delta synthesizing an Okazaki fragment to the 5' end of already synthesized Okazaki fragment. It creates a short flap at one or two nucleotides which is incised by Fen1 bound pCNA. So pole delta fills the short gap extending the nick and these events repeat by which RNA primers will be removed by creating short flaps. So DNA ligase finally seals the nick which is called as nick translation. So when large flaps are created, it will be bound by RPA that prevents FEN1 action. In such cases, an endonuclease DNA to trim the flap to release the RPA that enable FEN1 incision. So the group of cellular enzymes involved in this Okazaki fragment maturation and the last stages of elongations are include FEN1, that is flap endonuclease 1, and RNase H. The enzymes FEN1 and RNase H remove RNA primer at the start of each leading strand and at the start of each Okazaki fragment, leaving gaps of unreplicated template DNA. So once the primers are removed, a free floating DNA polymerase lands at the 3' end of the preceding DNA fragment and extends the DNA over the gap. So in the final stage of DNA replication, the enzyme ligase joins the sugar phosphate backbones at each nick site. After ligase has connected all nicks, the new strand is one long continuous DNA strand and the daughter DNA molecule is complete. So this is the schematic representation of the last Okazaki fragment maturation and the final stage of elongation. So for termination, there are no specific termination sites. They will occur when the internal replication fork fuse and the terminal replication fork reach the end of the duplex. So these events will eventually dislodge a replisome machinery, and you can see the image, the origin with synthesized leading and lagging strand on both sides. Replication of the lagging strand results in formation of an RNA primer at the end. Since the DNA polymerase can only synthesize in 5' 3' direction, the removal of primer will result in a gap that cannot be filled. You can see from the image that removal of primer results in the creation of 3' overhang. So it shortens the DNA strand, creating a 3' overhang, and this problem is called as end replication problem. If left unrepaired, the end replication problem may result in some of cases that can be daughter duplexes become progressively shortened after each round of replication, and critical genes in these locations are cut, resulting in severe mutations, and exposure of single-stranded DNA activate recombinational repair, resulting in fusion of chromosomes. Severe chromosomal aberrations may occur. So these are the major problems can be occurred when the end replication problem left unrepaired. The end replication problem is solved by the incorporation of telomeric DNA at both ends. Telomere is a G-rich region. In humans, it is a repeat of TTA-GGG. So these sequences are added on to the 3' overhang by telomerase enzyme. When the size of the telomere is around 5 to 15 kb, telomerase will dissociate and lagging strand machinery will synthesize a short primer and extend it using telomeric DNA as template. So this prevents critical genes from being cut, and it still creates a 3' overhang after the primer removal. So it is usually a 50 nucleotides long. This single-stranded overhang is looped and invade the duplex region of telomere. So they form a quadruplex DNA, and the loop is called as T-loop, that is telomere loop, that is G-rich. And formation of T-loop will displace the C-rich strand, that is newly synthesized strand, to create a small D-loop, that is displacement loop. So it prevents the exposure of single-stranded regions, and various proteins are required to extend the telomere form, that is to form D-loop and stabilize it. And these proteins involved are, many proteins are involved. Proteins involved are telomerase. So telomerase is a reverse transcriptase enzyme which is a RNA-dependent DNA polymerase. It is a ribonucleoprotein that contains RNA plus protein. So it is a complex of protein with bound RNA. It has two components, mainly TERT and TERK. TERT means telomerase reverse transcriptase component, which is a protein, and TERC is a telomerase RNA component, which is an RNA. So TERC, that is an RNA component, contains 159 nucleotide long, which is a short RNA sequence that is the rich region that act as template for reverse transcription, whereas TERT, that is protein component, contain four domains: telomerase N terminal domain, reverse transcriptase domain, then telomerase RNA binding domain, and C terminal extension domain. So, coming to the steps involved in this end replication problem, sorting, that is using the C-rich region in TERC, that is RNA component, telomerase hybridizes with 3' overhang. So the unhybridized region of TERC is used as a template to extend 3' overhang. So RT, TRBD and CT, these are the three domains in TERT, and these three domains in protein component in the telomerase together form a ring-like structure and close around DNA. So when the synthesis of RNA-DNA hybrid is complete, the ring opens up, splitting the RNA-DNA hybrid. So the telomerase translocates and hybridizes with the 3' end of newly extended DNA, and the reaction will repeat until the size of extension reaches the 5 to 15 kb, and telomerase will lagging strand dislodges, and the lagging strand machinery synthesizes complementary strand. And in the image, it's the last stage shown, that is the synthesis of complementary strand by lagging strand machinery. So this mechanism of synthesizing DNA by using the same template more than once is called repeat addition processivity. So T-loop formation and stabilization is done by a multi-protein complex called shelterin complex. Shelterin complex regulates telomerase activity and thus regulate telomere length. So there are 6 proteins in shelterin complex, which are TRF1, TRF2, TIN2, POT1, RAP1, TPP1, etc. So we have already uploaded a shorts with the proteins involved in shelterin complex in detail. So please watch that video if you have not watched it yet. Telomerase is active only in germline cells, stem cells, and tumor cells. So replication in somatic cells leads to progressive shortening of telomere. When the size is too small, T-loop cannot be formed. So protection of telomeric ends lost. So that this mutation rates increases and aging starts. So cells stop dividing. So the number of times a somatic cells can divide before they enter replicative senescence is called as Hayflick's limit. So the Hayflick's limit of normal cell is 50. And also, when comparing leading and lagging strand also incorporate telomeres. But telomerase cannot act on blunt ends, so a separate 5' 3' exonuclease will degrade the 5' end of leading strand and create a 3' overhang in the leading strand. And it is further processed by telomerase as same as in the case of lagging strand. So that's all about replication, elongation, and termination in eukaryotes. And we will be discussing about nucleosome assembly during replication and mitochondrial DNA replication and rolling circle model replication in the coming video. Hope you all understood about replication in detail. If you have any doubts regarding this section, please let us know through the comment section. And if you understood this video, don't forget to like and share to your friends. And please subscribe our YouTube channel, Like His Academy, your educational catalyst. Thank you.